Slides today. You guys know where the slides are. You know how to access them. I've extracted what I want you to know out of the slides. I'm going to say it out loud. All right? If you want me to pull up a specific slide, I do have it up here on the screen and we can do that. But I want to try to avoid getting into all that unless we're talking about a specific process such as a Calvin cycle or something like that where I may pull down the slide for that. For pure read and wrote, we're going through it. Okay? All right. So, on January 15, 2019, your first class, for I think all of you actually, your first class, we talked about some very basics. Do I think you'll see these on the exam? No, probably not, so I'm going to rat-a-tat-tat through them. But be aware of what they are in case you do. So what is the definition of science? Science is a body of knowledge, a collection of unified insights about nature. What are unified insights? Theories. That you might want to keep in the back of your head, the difference between theory and fact. A theory is a general set of principles supported by evidence that explain some aspect of nature. You will see a question on this. Be aware of it. Remember the scientific methodology, the steps that one has to take. Very quickly, it would be to observe, to question your observation, to hypothesize, to run an experiment, and either to conclude with evidence, one way or the other, or to re-hypothesize and run the experiment again. Okay? You probably will see a question about that where something will be missing or something will be out of order. So just remember the steps that you have to go through for the scientific method. What is a hypothesis? A hypothesis is a tentative, testable explanation that has not yet been proven true. Or false for that matter. It's not been proven either way. It may be tested through observation or experiment. Remember, not so much in biology, but scientific observation, well, we do observe in biology, but much more so ethnomethodology in sociology or otherwise are forms of observation. We tend to be a little more empirical in biology, but there are certainly elements of our experiments that are that of observation. So what is an experiment? An experiment is a series of actions done in a controlled environment to examine the validity of a hypothesis or the invalidity of a hypothesis. It must be open, it must be repeatable, and it must be falsifiable. So an example would be, as we saw, well, probably just as you guys were being born, most of you, Dr. Andrew Wakefield hypothesized and experimented to prove that there was a correlation between the MMR virus and autism. He actually came out with a whole study about it, yet because it was falsifiable, because it could be proven, not repeated, it was actually proven to be untrue. And it has been generally a debunked theory at this point. So that would be an example of the ability to repeat somebody's test or observation. Don't forget, take a glance at the slide to look at the Louis Pasteur experiment with the little flasks. Remember that? And spontaneous regeneration. Don't need to focus on it too much, but just understand that that was an example of something that a previous hypothesis, a previous theory that had been proven untrue as well. So what do different scientific disciplines share? I inferred that there was some sharing of observation, but they always share a use of evidence, a use of hypothesis, a use of logic, and a use of reasoning. Again, you should be able to tie that back to the overall scientific method or process of observing to hypothesizing, etc. So, here we go. Let's talk a little bit about biology. Can anybody tell me what the biology stands for? What is it the study of? Life. Very good. All right, do I think that'll be on the test? No, but be aware of what you've actually been studying for the last 16 weeks. All right, so this will be on the test in some way. Life is defined by 8 characteristics. Know them. All right, number 1, Life has the ability to assimilate and process energy. Number 2, something that's living can respond to stimuli or its environment. Number 3, something living can maintain or regulate an internal environment. Can anybody tell me what I mean by that? Homeostasis. I love it. Number 4, life, something that's living has the ability to reproduce. Number 5, has the possession of an inherited information base. What do I mean by that? DNA. Very good. Number 6, life can evolve from other living things. Number 7, life is composed of one or more cells. And number 8, living organisms are highly organized compared to inanimate objects. Okay? We are a little more diverse than a rock. Although rocks are impressive, you can ask any geologist. Know those guys. You will see something in that regard. I don't have an abbreviation or any way to remember it. Just think about it and try to focus on it, okay? Happily. Life is defined by 8 characteristics. Number 1, the ability to assimilate and process energy. Number 2, the ability to respond to the environment or be stimulated by the environment. Number 3, the ability to maintain or regulate its internal environment. Remember the slide about the rabbit? Big ears? Yeah? Okay, that's a form of homeostasis. Number 4, living organisms have the ability to reproduce. Number 5, living organisms have the possession of an inherited information base or DNA. Number 6, living organisms evolve from other living things. Number 7, living organisms are composed of one or more cells. And number 8, living organisms are highly organized compared to inanimate objects. That gets back to cellular structure, organ systems, etc. Okay? So we talked about homeostasis. Homeostasis means literally a steady state. The ability of an organism to maintain constant internal conditions, for example, thermal regulation, as I referred to the rabbit, or we can think about the African elephant with the larger ears. Okay, those would be forms of thermal regulation or forms of homeostasis. All right, next thing that you probably are going to see a question on, just one, but you'll see it. Life is hierarchical. What are the steps? What would be the very smallest thing that we acknowledge? Little teeny tiny thing that you can't see. Who said it?
Atoms.
Very good. Atoms, molecules, macromolecules, organelles, cells, tissue, organs, organ systems, organisms, populations, communities, ecosystem, and biosphere. Now we're going to stop on a couple of those. Yes. Don't raise your hand. Yell for me.
Oh, who comes after macromolecules?
Okay, after macromolecules is the organelle, which is what we're going to talk about right now, the organelle. All right, now where you're most likely going to get tripped up on this question is this. Remember that the organelle is not in order of itself. It's actually an organelle is within a cell. Okay, remember that an organelle is before a cell. All right, in the hierarchy. You should be able to go through organs, organ systems, organisms. The other one that you're going to see and make sure that we need to understand the difference between a population and a community. Okay? So I'm going to define those two and leave the rest to you. A population is a group of beings in a particular environment. Now, it's a little more technical than that, but that gives you the gist of it. A community is a group of populations in a particular environment. Remember the very most recent slide that we saw on ecology, where the elephant and whatever, the antelope and all of that in one single place? All right? That would be an example of a community. There were multiple populations within that group. Okay?
Yeah.
Yeah.
What was the name of that title you said for the order of like the
Like hierarchy.
Oh, okay.
I called it a hierarchy. I think I said hierarchical.
No, you're fine.
So, but it's a hierarchy.
Okay.
It's just a typology. I don't believe that he went over this, but then I think I saw it on the test, so I think it's therefore in your book. The taxonomical hierarchy, the old kingdom, phylum, blah, 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 okay? Again, I don't believe that he actually presented a slide on it, but I know it's inside your book, and I think it was on test 1 if somebody wants to kind of vaguely remember. So be aware of what that is. So it's domain, kingdom, phylum, class, order, family, genus, species. I'm not going through it. You guys should be able to jump on that yourselves. So, I will ask, what is reductionism? Reductionism is the attempt to reduce behavior to a physical level in order to explain it. An example of reductionism is when we study the brain, we study neurons and neurotransmission. We get down to the lowest common denominator. That's what you have to remember about reductionism. What is biology's unifying principle? I said it. Not to be confused with biology's central dogma. What is biology's unifying principle? What does the foundation of biology tell us? Evolution. We rely on evolution as part of our study and our analysis. Okay. All right, that's basically it. That was day 1, and it took us all of 12 minutes. Here we go. Now we get into the chemistry. My favorite part. Not really, I hate chemistry. What is a proton? Mandy, you've been writing papers too much. Come on now, you gotta be here. You drove all the way down from Fredericksburg. Didn't you? A proton is a positively charged particle that resides in the nucleus of a cell. A neutron, therefore, would be a neutrally charged particle that resides also in the nucleus of a cell. So we can probably conclude that an electron would be a negatively charged particle. However, unlike protons and neutrons, the electron surrounds the space around the nucleus of a cell. Okay? Can anybody tell me or remember what the octet rule is?
The electrons out in a shell wants to have 8 electrons in it.
Everybody hear that? Atoms of the main group of elements, i.e. up to carbon, tend to combine in such a way that each atom has 8 electrons on its valence or outermost shell. So when you're thinking shell, just a quick visual, think Saturn with the rings around Saturn. Okay, that's a quick way of thinking about it. Saturn is the nucleus. The rings around are the shells. The outermost shell is the valence shell. The octet rule says that the valence shell in the primary main elements, that is the elements up to carbon, want to have 8, such a way that each atom has 8 electrons, okay? And you know it's electrons because you know electrons hang out on the shells as opposed to inside of the nucleus, okay? We don't have a table of elements here. Although, you know, it's interesting. Is anybody over in Temple? Is any psych majors here?
Yeah.
Wow, there's a lot of you. Okay. So why do they have a periodic table of elements sitting there in that big forum that they use? That makes no sense to me. There's no chemistry classes. There's no biology classes over there whatsoever. Anyway, all right. So a couple quick characteristics that you can look at yourself, and I'm not going to pull down a periodic chart. But basically, the atomic number is the number of protons, and therefore, obviously, usually the number of electrons as well. The mass index is the number of protons plus neutrons. Okay? So therefore, a way to determine the number of neutrons in an element would be to subtract the atomic number from the atomic mass. All right? It's a little complex. It's not worth going over, but you probably will see one question on the periodic chart itself. You got to kind of glance at it. Look at the difference between atomic mass versus atomic number. Okay? What is an isotope? Oh boy, you guys have been gone for 16 weeks. You got to come back to this. There's going to be 20 questions of this stuff. An isotope is an element that contains the same number of protons, but a different number of neutrons. A way to think about that would be to look at carbon. There's carbon 12, there's carbon 14, okay? Same number of protons, but a different number of neutrons. What is an ion? Keep going. An element with an unequal number of protons and electrons to create the charge. A positive ion is called a, and a negative ion is? There you go. Everybody hear that?
No.
Do it again.
A positive ion is a cation, and a negative ion is an anion.
Okay? All right. These you will have to know thoroughly, not what I just covered. That's why I went fast through it. These you will have to know thoroughly what I'm about ready to do. There are different types of bonds. Okay? We need to think about those different types. We have the ionic bond. Anybody get one for me? Say it.
I said covalent.
Covalent. Okay. What's another one? DNA uses it?
Hydrogen.
Hydrogen. And what's the one that's rare and named after somebody? Van der Waals, which is really a form of hydrogen bonding in and of itself. So an ionic bond, what I want you to think about is that it's a lock and key, where the electron is donated to the balance of another element connecting it. Okay? So therefore, by definition, if we think lock and key, we can assume that, well, we can hypothesize for ourselves that this is the strongest type of bond. So an ionic bond is the strongest bond.
You said another what?
Say again?
You said the electrons donate to the balance of another what?
Yeah, to the balance of another element. So basically, I put my key in the lock, and then the key is there. I brought the connection through. The second, the covalent bond, is where an electron is actually shared between two elements. The third, which we refer to as hydrogen bond, which we would use DNA as our example, or at least the interior part of DNA, not the helix itself. And it is a chemical bond between two polar covalent bonds. Remember, the hydrogen bond has to use polar covalent bonding to begin with, creating a slightly positive and a slightly negative charge. And then Van der Waals is the weakest, and it's simply a chemical bond between polar atoms in different molecules. I would suspect that you will see something regarding the polarity necessity in hydrogen bonding. You should think about water as your example. All right, what is an element? Any substance that cannot be reduced to a simpler set of constituent substances by a chemical means. So, when we look at the periodic chart, those are the lowest common denominators. They're not getting any smaller than that, to the best of our knowledge. Can somebody explain to me the difference between a polar and non-polar covalent bond? Polar bonds form at one axis or another. So you think about your Mickey Mouse ears. Remember the polar bear example that he had up there? And he referred to the ears. I like to think about Mickey Mouse. But basically, all of the bonding happens on one pole or the other. Nonpolar by definition then, would be where they're evenly formed around the element. Okay? I inferred this, but I want to be very clear. You will see it. Polar bonds are the only type of bonds that can create or form chemical bonding after that, hydrogen or van der Waals. Okay? Only polar bonds. So, I used as an example, and we should know this, what type of bond would water be? But it has to start with what? Polar. It's a polar covalent bond. Correct. Remember that it's polar to start. Why is water so important to life? You will see this. You need to know it. Water is a solvent. Water has high density, yet it has less density as a solid, thereby allowing life to persist. And water has a high specific heat. It serves as a thermoregulator for our biosphere, Earth. Okay? So, solvent we'll get to a little bit more when we get to base and acid. But understand the concept that if you laid in the bathtub into perpetuity and were fine otherwise, that you would eventually dissolve. Water is a solvent. Okay? Number 2, water has a high density. We'll need to know this as we get into, as we get into other elements. But what I want you to remember there, what I think is important, is that when it becomes a solid, it actually is less dense. And because of that, we think about the fishies underneath the pond being able to live in the wintertime while everything else is frozen over. Life can persist. That less dense state allows oxygen to get through, allows some degree, some light to get through, etc. Okay? And water has a high specific heat. Don't forget that 90% of our Earth is surrounded by, is covered with water. It has a high specific heat allowance, therefore it serves as a thermoregulator for the Earth's crust and the Earth's core. Okay? Any questions? Can anybody tell me, I call it the solvent equation. Give me an example of solvent. I just said it. Okay, give me an example of what would you put in water? Salt. What would that be called? Solid. Good, solute. And once that salt is all mixed up and you have wonderful salt water, but there's no more grains of salt explicitly, that would be called the excellent. Some of this stuff I've already hit. Talk over my own questions. Why? Do that. Does water have a high or a low degree of cohesion? Well, actually, let's stop. What is cohesion? Anyone? You will see a question on cohesion and adhesion. Pretty much guaranteed.
Ability to stick to itself.
Cohesion, I want you to think about exactly that. The ability for water to connect to other water molecules. Think about a glass of water. Keep putting drops in it. In theory, as long as you do it carefully and minutely enough, and that's where you get the little dome on a glass of water. The water cohedes to each other. What is adhesion? Adhesion is, think about the straw. We get into it when we're dealing with the xylem inside of plant life. Okay, adhesion brings it up. Okay, so what I want you to think about there is the attraction of water molecules and other molecules, i.e. the straw. Okay, so it isn't just the cohesive property, but it is the adhesive property, the ability to attach to something. All right, somebody want to tell me hydrophobic and hydrophilic? Phobia is afraid. So there you go. So hydrophobic is the inability to mix with water. Think afraid of water, whatever it is. So when we put our oil, when we're heating up pasta, and we put our salt in, the salt does very well. But then when we put our oil in, or a little bit of oil, olive oil in, it does not mix. Okay, it is hydrophobic. All right. So I've just given you hydrophilic, the salt within the pasta, water, and hydrophobic. All right. We're moving right along. Can anybody briefly describe to me the pH scale? You'll probably see a couple of pH scale questions on there, okay? So be aware of it and take a glance at it. I have my little teeny tiny one, but it's in the slides, all right? I would definitely take a glance at it and know a few basic fundamental pieces of the pH scale and how it works, all right? So, if we know that a pH scale goes to acidic or base, all right, we know that the middle, which is roughly 7 on the pH scale, would be water. Water. And just as a side note, what's awful close to water as far as it being right there in the middle? Neither acidic nor base. You all have it. What? Spit too. Spit with turbid water. All right, if we take a glance at it, all right, know that a couple of things. Know that it is a base 10 scale. It goes from 0 to 14. 0 is the most acidic. 14 is the most base, or basic. 7 is considered neutral. Blood is very close to that neutrality. So let's get into a little technical stuff, a little chemistry. What makes something more acidic? If something becomes more acidic, it's because acid is yielding hydrogen ions. Very good. Very good. What makes something more basic then, therefore, is the base accepts hydrogen ions. Okay? So, because I've already asked you several times, at what pH balance do most living things function? Number 7. 7, right in the middle. Okay? Think blood being right there. It's actually not quite 7. Think water. We're all made up of water, blood, etc. And we are excellent representations of living organisms, or at least some of us are. All right. Water cycle. Everybody have the gist of that? Do we need to really go over it? It rains, goes in the dirt, it evaporates, goes in, blah, 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 blah. Okay? I'm not going to go over this thing. Because we have like 20 pages to go on this section. What are the 4 functional groups? Hydroxyl, carboxyl, amino, and phosphates. Hydroxyl would be carbohydrates. I know that doesn't make sense because the next one says carboxyl is actually fatty acids. Amino, we should be able to figure out, are amino acids. And phosphates would be 2 major things that are phosphates. You know them if you've taken any exams. One we've already talked about today. The double helix is a phosphate. Okay, so DNA is a phosphate. But the other one is, what's the highest form of energy? ATP, adenosine triphosphate. Okay? So high energy and DNA are types of fall in the functional group of phosphates. What is a monomer? A monomer is a single molecule that is able to bond in long chains. So therefore, what is a polymer? It is the long chain, a bunch of monomers put together. Okay? Should be able to alphabetically, if somebody were saying M-N-O-P, P's later, so it's bigger. What does hydrolysis do?
Releases water.
Huh?
Releases water.
That?
Releases water.
Yes. Very good. Well, it adds water to the polymer bond. Okay. Dehydration is where it removes water. Sorry, I went backwards on that. So again, because I messed up, what does hydrolysis do? This is the occasion where water molecule breaks a polymer bond. Okay? Dehydration, and you can think about this if you're outside or like me, soaking wet already up front here. I'm getting dehydrated. I'm losing water. Okay? So so dehydration removes a water molecule to form a polymer bond. All right, I'm going to quickly go through, but I would like you to know this. 3 examples of a monomer and its related polymer. Monosaccharide becomes polysaccharide. Amino acid becomes polypeptide. And nucleotides become one of 2, DNA or RNA. Okay? Carbohydrates are monomers of simple sugar. Another word for simple sugar, the proper word is glucose. Good. That link to form polysaccharides, which are considered complex carbohydrates. Okay? What are examples of carbohydrates? These you got to know these guys, and they're painful. Glycogen, cellulose. One more.
Chitin.
Very good. Chitin. What are lipids? Well, first of all, are lipids hydrophobic or hydrophilic?
Hydrophobic.
Very good. Think of the olive oil. They form fats, oils, and waxes. What is the lipid polymer structure? Trick question. There is no commonality in lipids. They don't form a specific structure. What are the 4 groups of lipids? Again, I want you to know it. Glyceride.
Steroids.
Steroids. Very good.
Hormones.
Huh?
Hormones.
I'd play that out on steroids, even though it's not technically. Phospholipids. And the final one would be waxes. Okay? What are triglycerides? All you got to do is look at my big belly. Fat consumed by animals to act as an energy storage. What are some examples of steroids? You said it, but break it down even further. And very good. And cholesterol would be the third. So cholesterol is actually a form of a steroid, as well as testosterone and estrogen. Okay? What are phospholipids? Well, what's an example of a phospholipid? We're all wearing it. Okay, so when you think of skin, you have glycerol and phosphate that make up an outer membrane. Okay? We'll get a little bit further into phospholipids under plants. But don't forget that which part of the phospholipids, everybody knows what a phospholipid looks like, right? Yeah, kind of like a stick person, okay? So if this is the tail, and this is the head, telling on an art major, which one is hydrophobic and which one is hydrophilic? The head is what?
The head is hydrophilic.
Head is hydrophilic. Very good. And the tail is hydrophobic. All right? Very good. What are waxes? single fatty acid linked to long chains of alcohol. Don't forget the alcohol part. What do waxes perform? Think about your plant leaf. They act as a sealant or barrier. They act as a sealant. So for example, waxes are on the epithelial tissue of a plant leaf on both sides other than the stroma, and they act as a barrier, only allowing in what they want and protecting the plant or the leaf from anything else. What are proteins? Proteins are molecules composed of monomers, and they are called, very good, that was what I was looking for, amino acids. So what would the link of amino acids be called? Polypeptide chains. Very good. That's what you said, right? Or you were close. Okay. All right, so what are the 4 structures of proteins? Yes, primary. So therefore we know the next one would be,
Secondary.
and we know the next one would be,
Tertiary.
and the final one would be,
Quaternary.
very good. 1, 2, 3, 4. All right. I would suggest you glance at that. One looks like, well, I could actually draw it out real quick. So 2, 3, 4. So if this is primary, secondary, tertiary, quaternary. Primary would be, think about a little, just a single chain, okay? You with me? Okay, by the time it becomes secondary, that chain has actually been folded, or has the ability to be folded, or, as we know about a helix, it could be, which I'm not going to try to do, but something like that. It has a specific pattern in the secondary. In the third, it starts to combine with other ones, which makes a big gobbledygook. And then fourth, it sets itself in. Okay? Know what the 4 basically are. What are nucleic acids? Polymers of nucleotides. Good. Can anybody tell me what 2 nucleic acids are? I just said it. DNA
And RNA.
and RNA. So DNA is deoxyribonucleic acid, and RNA is just taking the oxy off. We'll get a little bit more into DNA, obviously, in various portions, but let's not forget that nucleotides contain a sugar, a phosphate, and 4 nitrogen-contained bases. Okay, the sugar and the phosphate make up the double helix. And the 4, can you name the 4? Can anybody name the 4? Adenine, guanine, thymine, and cytosine. And what gets replaced in RNA? Thymine comes out and uracil comes in. So therefore, let's get a little bit into DNA. What is DNA? DNA is the genetic information of a cell. The base sequence encodes information for the production of proteins produced by living things. What is, unlike its primary principle, what is biology's founding, central dogma? Very good. DNA to RNA to protein. Know the order. We'll get into it further in another section, but know it. With one exception, that will be my follow-up question, what is life's fundamental unit? Don't say atom. Cell. What would be one form of life that doesn't have that? Viruses. That's why we don't consider viruses. To be living organisms. What is a prokaryotic cell and what is a eukaryotic cell? right? You ready? Yeah.
Prokaryotic is like bacteria, right? And eukaryotic is just like living things.
Think more prokaryotic is a simple single-cell organism without a nucleus. And the eukaryotic is the more formed, has a membrane-bound nucleus and organelles. So, probe or before would be an earlier form of a cell, even though they both exist. I would like you to make a note to yourself to look at the slide on an animal cell and be able to basically cover the important pieces of it. Plasma membrane, the mitochondria, cytosol. You don't need to get so far into all of this, but there are some basic things that you need to be able to explain anatomically of a cell. What is the function of a nucleus? What does it hold or house? And therefore, a nucleus also directs the synthesis of ribosomes and proteins. What is the nuclear envelope? Nuclear envelope is the double membrane structure on the outermost portion of the nucleus. What is the ER? What does ER stand for? Don't say emergency room. Endoplasmic reticulum. What are the different types of ER? So much mumbling, you guys start talking. Rough and smooth. Rough ER is the endoplasmic reticulum upon which ribosomes are attached to create proteins. The smooth ER is on which the synthesis of carbohydrates, lipids, and alcohol occur. What is the Golgi apparatus? Think of a publisher. When you think of the Golgi apparatus, think of a publisher or an editor. It's the final editing of the information before it goes out for use or for purpose. Okay? So it takes a protein, it edits it for a very specific purpose, and then it sends it out to go do its thing. What is the general, if I were to say quickly what the general process is of protein synthesis, could somebody do it for me? First of all, there has to be an instruction to the nucleus. And then what happens in the nucleus? Something unzips. DNA unzips, and out comes RNA. RNA attaches to the ribosomes on the... Which one? Rough ER, good. And that is where the formation of amino acids begin. And from the amino acids, remember, polymers, monomers, from the amino acids, proteins start to generate. The formed protein then goes on to the Golgi apparatus for final editing, and then gets sent out to do its thing. What are lysosomes? This is the cell's garbage disposal. So with digestive enzymes, proteins, polysaccharides, lipids, nucleic acids, and worn down organelles are removed. All right? Don't try to pay attention to the lysosome. I have a feeling it'll show up. What are vesicles versus vacuoles?
Vesicles are specific things traveling in water.
They're both storage and transportation. You're correct there. But make sure that you emphasize that vesicles are smaller, vacuoles are larger sacs. The other important thing about the difference between vesicles and vacuoles, just to be aware, in case you see it, is that vesicles can fuse between cells. Vacuoles are too large and cannot fuse between other cells. But that's good, you're exactly right as far as what they both do, they both do the same thing. Just make sure you can separate and synthesize the difference between the two. Okay? Let's go back for a quick one. What are ribosomes? Ribosomes are responsible for protein synthesis. They exist on the rough ER. Very good. What is mitochondria? Mitochondria are the energy producers of a cell, and they make ATP, which stands for adenosine triphosphate. Mitochondria is double membrane. It has its own DNA and ribosomes. What does a plant cell have that an animal cell does not? Very good, perfect. What is the cytoskeleton? Well, what is the cytoskeleton made of? Protein. Cytoskeleton is a web of protein strands that are the structure of a cell. You can think bone, okay, within a cell. Cytoskeleton, however, just like bones do, assists with the movement of a cell and the movement of organelles within the cell. What are the three principal types of cytoskeleton elements? Microfilaments, intermediate filaments, and you got it. Microtubules. Very good. So microfilaments function in the movement of cellular components. So they're not helping with the movement of the cell, but cellular components, organelles within. Intermediate filaments are the structural function of a cell. That's the true bone, as it were. And the microtubules actually guide organelles movement within. Okay? Do plant cells have lysosomes? No. Do animal cells have a cell wall? No. Do animal cells have chloroplasts? No. Do they have a central vacuole? No. What is a central vacuole? It's the fluid filler within a cell, pushes organelles together. Remember, when we get into plants, we'll be studying that. So if I were to talk about the differences of communication between plant cells and animal cells, how do plant cells communicate? Plasmodesmata, membrane-lined channels, and the cytoplasm is connected. Versus animal cells. Animal cells have adjacent cells, have channels called gap junctions. Very good. You'll see gap junctions somewhere, somehow. Whether it comes up as an answer or part of the question, I couldn't tell you. Animal cells allow movement of small molecules and, pointing to it, what fires neurons, neurotransmission, electronic impulses happen within animal cells. Also remember that there is no membrane lining, just a tight junction between different cells. What is passive transport? Almost done. What is passive transport? Passive transport is where no energy is spent by the cell to accomplish movement. What is diffusion? Is diffusion a form of passive transport? Yes. But diffusion does something very specific. Diffusion allows movement from a cell from low to high or high to low concentrations. Always high to low. What is facilitated transport? Movement with the assistance of transmembrane proteins. Another word for transmembrane proteins that we could use. What helps you get up the hill? Enzymes. Assist with that, remember? What is osmosis? Well, we just said it. Osmosis is a form of diffusion, so therefore it's moving water from a high to low in order to even out the, what was the middle one? Not the solution, but the solute. Okay? Remember the example? I don't know that I have a good one here, but remember the example of the half a glass of water with the membrane line there. Dump a lot of salt in one side, only a little salt on the other side. Assuming that diffusion or osmosis can happen, it wants to even out the solute. Okay? The water's going to stay the same no matter what. It's not going to rise or fall, but it wants to even it out. What is tonicity? The amount of middle stuff. I just said it. Solute. Tonicity is the amount of solute in a solution. What is osmolarity? The measurement of tonicity in a solution. What is a hypertonic solution? A hypertonic solution states that the fluid outside of a cell has a higher concentration of solutes. What is an isotonic solution? Even. And a hypotonic solution would therefore be that the fluid inside has a higher concentration. Remember those three. Hyper outside, iso middle. You can work your way back to hypo. What does active transport require? What kind of energy do I love? ATP. What is endocytosis? The type of active transport that moves particles or cells into another cell. Just remember that endocytosis is active. What are three types of endocytosis? Guarantee it's going to show somewhere. Phagocytosis, which is large particles. Pino, which is smaller particles being taken in by a cell. Anybody come up with a third one? Receptor mediated. Particles bind to proteins on a plasma membrane, and the membrane invaginates or depresses, bringing the substance into the cell. For my gross layperson purposes, I think about a pimple being covered up on your face. Okay? What is exocytosis? Go back to the pimple. The receptor-mediated pimple has now been brought in. You as a human being don't want the pimple on your face, so you privately go to the bathroom, or some gross individuals maybe publicly, and you pop it, and you have now expelled the material from within the cell into the outside.